Okay. Yeah, so hi everyone. I guess just let me know if you can't hear me. But yeah, I'm Leah. I'm a student at Penn, where I work with Danny Bassett in her complex systems group. And before I start, I just wanted to say that Danny was originally supposed to be giving this talk today on some work the group has been doing on large-scale brain states. But she unfortunately had a conflict and so asked if I could fill in for her since I was supposed to be in Denver for APS. So I just wanted to apologize a little bit ahead of time that the talk that I'm able to give today maybe is more tangentially related to the topic of the workshop session. I think maybe what's going to be different about the talk that I'll be giving is that it's going to be a purely simulation-based study examining the effects of perturbations in a computational model of large-scale brain network activity. And what we're specifically interested in asking is whether the impact of perturbations depends on the collective dynamical state of the system, which I think is a notion that maybe hasn't been fully appreciated or explored in the context of macro scale brain networks. So although I think maybe this talk will be of kind of a different flavor, I'll also try at the end to briefly note some ways I think the analysis approaches that are being discussed today might be able to help us improve upon the kind of mean field modeling framework that I'll discuss. Okay, so let's see. So yeah, this is just a brief outline of what I'm going to try to present today. So start with a little bit of background and motivation, and then I'm going to introduce the model of large-scale brain activity that we implemented. So this involves combining human structural brain data with the Wilson-Cohen neural mass model, and then I will present some results. So first, I'll briefly discuss how we can in a simple manner, change the regime or the collective state of the model by tuning a single parameter in the equations while we leave structural network connectivity fixed. And then we'll focus in on two distinct states of network activity. And for each case, we'll examine the effects of focal perturbations on system dynamics. And I'll mainly focus on how localized perturbations can modulate functional interactions in the network, and hopefully we'll see that the effects of perturbations can differ significantly depending on the state of the system. Okay, so just going to start with a bit of background and motivation. So for this project, we want to construct a model that allows us to study the effects of perturbations for different states of network activity. So to start, I just wanted to sort of briefly outline a few basic points that kind of guide how we're going to set up this model and how we're going to think about this question. So as maybe you already know, Danny's group focuses mainly on large-scale brain organization and dynamics. And at the macro scale, we can really think of brain activity as arising from a complex, spatially distributed network of many interacting regions. And we also know from empirical measurements that at the scale of these large neuronal ensembles or brain areas, that population dynamics are often rhythmic or oscillatory in nature. And furthermore, this rhythmic activity can become temporally coordinated across the network and yield very complicated spatiotemporal collective dynamics. And we think that coherence of these neural oscillations facilitates a number of cognitive processes. So sort of due to the ubiquitous and very important nature of large-scale oscillations and their interactions, there's in general been a lot of work trying to understand how the organization of brain network structure shapes system-wide dynamics. The related question that we're specifically interested in here is how does a change of neural activity in a local brain area interact with ongoing dynamics and potentially reconfigure these kind of long-range functional interactions? And how does the modulation of those interactions depend on the structural network architecture, but potentially also on the state of network dynamics? So just for a little bit more motivation here, I think it's now sort of quite well established that population level oscillations can be induced or modified by different kinds of perturbations, whether that be sort of natural sensory inputs or more artificial perturbations like brain stimulation. And in general, there's currently sort of a lot of translational work being done that tries to use these kinds of focal perturbations of neural activity to first gain insight into healthy brain function, but to potentially control or alter abnormal dynamics. So I think that's one big motivation for trying to understand the influences of perturbation or stimulation. However, I also think that there are sort of a number of open basic science questions regarding how perturbations affect brain activity. And so one big challenge that Danny's group has been trying to address in recent studies is to understand not only the local effects of perturbations, but also how those effects can propagate in a larger brain network to modulate dynamics at a collective scale. So how does a perturbation of a single brain area interact with ongoing oscillations in the network to potentially change functional or temporal relations? And then a second sort of related and important question is whether the impacts of perturbation are determined solely or mostly by the anatomical network, or if they also depend on the state of network activity. So I think that in sort of large scale network neuroscience, there's really been a lot of focus on trying to predict patterns of dynamics from the architecture of these structural connectomes, which has been sort of a very fruitful way of trying to understand large scale activity patterns. But we also know that macro scale brain dynamics can exist in multiple states, even though these white matter networks have relatively fixed connectivity. And so a single connectome can give rise to sort of a diversity of collective states. And a question that we're particularly interested in is whether the impact of perturbations depends on that state. But I think sort of one challenge that hinders progress regarding both of these points is that it's still very difficult to go in and systematically and exhaustively examine the network effects of selective perturbations to several different brain areas. It's difficult to do that empirically, and it can also be hard to control brain state in a way that we understand or have a good grasp on. But we can use in silico or computational approaches to start getting at these questions. And so what we wanted to do was build a dynamical model of network activity, brain network activity, to start sort of conducting a basic first examination that would allow us to simultaneously assess the distributed and the state-dependent effects of perturbations. Okay, so we're going to construct a simplified model of brain network dynamics using a relatively common approach in which large-scale activity is modeled as arising from a network of coupled neural masses. So in this type of framework, the first step is to represent or to think of the brain as a large discretized network of interacting dynamical elements. And so to do this, we're going to take brain imaging data, and then we're going to parcellate the brain into distinct spatially distributed regions. And then we're going to construct a network by connecting those nodes or those regions with edges that are going to represent white matter streamline counts. So these anatomical pathways are derived from empirical diffusion imaging data and are going to determine at the large scale how the dynamics of different areas in the network are coupled. So the figure that hopefully you see on the far right depicts a group averaged structural human brain network projected into two-dimensional real space. Okay, so the next step is to specify the dynamics of each region. And here, the typical approach is to use a reduced neural mass model. The reason being that it's impossible to sort of build a model at the level of single neurons if we want to try to understand the dynamics at the scale of the whole brain. So here, we're going to represent each node in the network as a Wilson-Cohen unit, which is this well-known mean field model of neural population activity. And importantly, this system can generate oscillations. So there's this bifurcation parameter, the excitation level PE, that controls the onset and then the strength of rhythms in an isolated unit. And later, this bifurcation parameter will also be the quantity that we tune to modulate the state of network activity as a whole in this controlled manner. Okay, but first, I have to explain how we can scale this up to a whole brain model, which is the goal here. And so to do that, we're going to take our individual regions from our parcellation, and we're going to couple them according to the topology of this empirical anatomical brain network. So Wij here would represent a long-range structural coupling between two areas, and Tauij is a time delay between them since they are spatially distributed. And if we do this, then we get a modified system of equations where we now add a term corresponding to these delayed network interactions. So in this kind of approach, large-scale brain activity is modeled as a collection of these oscillatory neural masses where the long-range interactions are informed by structural data. And it's, I think, important to note that this modeling framework that we're sort of implementing and building off of here is based off a number of past efforts that have tried to use this networked neural mass setup to investigate relationships between brain connectivity, structural connectivity, oscillations, and in particular, synchronization of neural oscillations. Okay, so we're interested here in perturbations, the effect of perturbations. And so we need to incorporate that into this model. And we're going to start by doing that in the simplest way, which is to just increase the level of excitation to a single region, say region J, while holding the excitation fixed at its baseline value for the rest of the network. And then because this is a model, we can repeat this perturbation process for different choices of the stimulated site, and then we can study how the network responds as a function of the stimulated site. Okay, so we're interested in asking if the state of dynamics affects the outcomes of perturbation. So I'm first just going to show briefly that we can change the baseline dynamics of this model by tuning a single parameter while leaving structural connectivity the same. So start by simulating this system in the absence of perturbation, but we globally vary the main Wilson-Cohen bifurcation parameter, which is the level of input to each unit. And so as with an isolated node, we see that varying this parameter clearly alters the dynamical state of the system, which here we can see by examining the nature of regional activity and its corresponding power spectra. And so in particular, as the global excitation level increases, the strength of the endogenous oscillations is enhanced. Okay, but in addition to just the activation level of individual network components, I think we also care equally as much, if not more, about the collective state of the network's dynamics as a whole. And here by collective state, I'm just going to mean the organization of the long-range dynamical coordination between different areas, although there are, of course, many ways to define what the state of one of these networked systems is. But to formalize the state in this way, we can just use the phase locking value to quantify the pairwise coherence of regional activity. So here, hopefully you can see on the lower part of the screen, the resulting pairwise coherence matrices for varying levels of this excitation parameter. And so what we see first is that even though the large-scale anatomical connectome remains fixed, the organization of the dynamical ordering in the system can vary significantly as we change the behavior of local oscillations. Okay, So we can sort of push the network into these different local and collective states by varying a single parameter in the model while holding connectivity fixed. And so what we wanted to do next was examine how system dynamics are modulated when one part of the network receives additional input. And what we were specifically interested in asking is if and then how the network response differs when the system is perturbed in state 1, characterized by these strong endogenous rhythms, versus in state 2, where there's still oscillatory activity at each region in the baseline state, but they're sort of much weaker. The spontaneous dynamics are weaker. So for the rest of the time, we're going to look at how the effects of perturbations, how they manifest in the network, and whether the effects depend on baseline state. Okay. So I wanted to start by looking at how perturbations alter regional activity in state number 1. So here on the left, I'm just showing the activity of one area in the network at baseline, and the right panel shows the activity for the same area when it is excited. And then to more clearly see what, if any, the effects of the perturbation are, I think it's helpful to study the spectra. So here in the lower left plot, the gray curve corresponds to the spectra at baseline, and the blue curve corresponds to the spectra under perturbation. And hopefully you can see that the effect of this kind of perturbation to the network is to slightly increase the amplitude and frequency of oscillations in the directly excited area. Okay, but what about downstream areas in the network that aren't directly stimulated? So Danny's group is specifically interested in network behaviors, and so we care a lot about moving beyond the local effects of these perturbations. And so here in the middle and right plots, I'm showing spectra of two other regions, K and L, where K is located very nearby to the area that receives additional input, area J, and L is located far from region J. And what we see, or maybe it's difficult to see because the curves overlap, is that spectral modulations to downstream areas activity weaken very rapidly for downstream areas. Okay. On the other hand, if we go back and we perform the exact same perturbation, when the network instead operates in state 2, where population rhythms are weaker, we see that the result is quite drastically different. So here, I'm again showing the activity of one area at baseline and when it's perturbed, and then the bottom left plot shows the spectra in those two conditions. And so as for the high activation state, we see that excitation shifts the amplitude and frequency of the oscillations, but for this state, the changes are noticeably stronger. And so to better quantify the difference in the sensitivity to perturbation at the regional level, we can compare the average shift in frequency, peak frequency, that's induced by a perturbation in state 1 and in state 2. And so I'm showing that average shift in the bar plot on the right, from which it's clear that the perturbation effects are significantly enhanced in state 2. And then furthermore, if we again consider the effect of the focal perturbation on downstream area spectra, we also see that for state 2, for this state, the effect of a perturbation can clearly spread in the network. So even though downstream region K, shown in the middle panel, doesn't receive direct excitation, the activity changes that occur in area J when it is perturbed propagate and induce these broadband modulations to area K's spectra. So when the system operates in a regime of these weaker baseline oscillations, both local and downstream activity is more flexibly modulated by a change in neural activity at a single area in the network. Alternatively, in a state of stronger endogenous activity, regional dynamics undergo much weaker changes due to an excitation of the same strength. Okay, So we wanted to follow up on this a bit further and ask not only how perturbation affects activity, regional activity in isolation, but also whether it leads to changes in the actual dynamic coordination between different network elements. And so to quantify this, we're just going to look at the pairwise changes in coherence, which I'll denote as delta rho, between the baseline state and the case when a single unit in the network is focally stimulated. So again, starting with state number 1, these two lower plots show examples of the delta coherence matrices when two different sites in the network are perturbed. And what we see is that even though the perturbation is very localized, it occurs in one node of the network, we observe these distributed changes in interareal interactions. And to just summarize the overall diversity in the network response, the purple bar plot on the screen shows the average absolute change in coherence, averaged over all pairs of regions, induced by perturbing a given area in the network. And so we see that these kinds of perturbations do also reconfigure the collective dynamics of the network, and different areas produce effects of varying magnitude. So given that we observe these changes in long-range functional interactions, our next question was, what aspects of the system might predict the overall variability in these responses? In other words, is there a significant relationship between the overall change in regional coordination induced by a perturbation and a network property of the perturbed site? And what we found is that for this state of the system, the structural connectivity, or the strength of the anatomical pathways that are linked to a given area, is indeed strongly related to the global change in coherence that occurs upon regional stimulation. So this tells us that when the system operates in a state of these robust regional oscillations, that strong network-wide effects of perturbations are really mediated by the presence of strong anatomical links from the perturbed site. And I think this tends to agree with our initial intuition that there should be a strong overlap between the anatomical network and a measure of the function of the system or the dynamics of the system. Okay, but what happens if we change the dynamical state of the network, we alter that, but we leave the anatomical network fixed? Then do these same perturbation-induced changes in relationships also hold in state 2 when regional oscillations are present but weaker? And so here, I'm again showing the delta coherence matrices when two units are excited, and then the bar plot shows the network averaged response for each choice of the perturbed site. And as before, we find that these perturbations can lead to distributed changes in the collective dynamics of the system, and there's again this heterogeneity in the response depending on which part of the network is activated. But what is particularly interesting is that for this state, structural network connectivity is not related in a simple manner to the global changes in network coherence that occur upon focal perturbation. And instead, what we find is that for this state, it's areas functional connectivity that's a significantly better predictor of the response. So in other words, regions that have more dynamical coherence or integration with the rest of the network to begin with are regions that lead to larger overall changes of long-range coordination when they are perturbed, which you can see in this second plotted relationship here. So what this, I think, allows us to begin concluding is that the effects and the mechanisms that govern how perturbations interact with large-scale network dynamics are not necessarily fully constrained in a straightforward way by the anatomical connectivity, but can also depend significantly on the state in which the system operates. Okay. So at this point, I just have a couple more slides. So first, I just wanted to quickly summarize. So I think first, trying to understand the effects of stimulation or perturbations in macro scale brain networks is an important area of study with potentially clinical applications. But really one of the only ways that we have to explore the effects of this in a systematic manner is to build, analyze, reduced computational models of large-scale neural activity. And so we implemented such a mean field model and found that the distributed effects of local perturbations depend definitely on the site of the stimulation, but also on the collective state or the working point of network dynamics. So even though anatomical scaffolding is fixed, it doesn't fully determine in a simple way the impact of perturbations, which I think is a point that maybe hasn't been fully looked at on the scale of the whole brain yet. Okay, so this is my final slide. And to conclude, I first wanted to note that this study, as I'm sure you can all tell, is obviously highly idealized. And there are, I think, a lot of ways that we can improve upon these kinds of large-scale models. And I think one of the most important steps is to try to constrain these neural mass models with empirical measurements of activity, brain activity, which could come from MEG or EEG. But I think one issue is that there are obviously many properties of network recordings that we could ask our simulation to match. And so, for example, it's common that researchers will try to match the structure of functional connectivity networks between data and simulation. However, there's also been some recent work on whole brain activity that uses dimensionality reduction techniques to show that at least sometimes large-scale dynamics can be well represented by lower dimensional manifolds, and these succinct representations can often be related to behavior. And so I think it could be interesting to try to use the results of data reduction methods that are applied to empirical data to inform how parameters in these large-scale computational models can be tuned such that the model recovers similar lower dimensional structure, which might be extremely important in guiding behavior. And then in the reverse direction, I think that we can also potentially use these large-scale simulations where we know the generating equations to help us understand what determines the output of different dimensionality reduction techniques or different data analysis methods. So for example, if we know that our model is a reasonable approximation of neural activity, then we can go in and we can systematically vary parameters, or we can change the network topology, and we can ask how those perturbations affect the result of our data analysis technique, which I think might help us make predictions or learn about what aspects in these large-scale brain networks give rise to potentially lower dimensional state-space representations. And that's all I have for you guys. Thank you so much.
Thanks, Lia, for the great talk. So yeah, is there anyone has a question with related talk? And you can come to the front to. Not really? Okay. I actually have a question. Hey, Lia. Oh,
yeah.
So my question is, yeah, you've been talking about this fitting to the, like, basically you build a network and simulated data from the network and just try to perturb to see the dynamical change. I think this is probably a little beyond the scope of this talk, but I'm curious, like, have you tried to fit this to the real data? Because this seems to be more like fMRI data, right? So have you tried to fit this to the real fMRI data, and how you kind of, how to say, because for fMRI data, the functions changes a lot as at different time points. The function itself, even without the perturbation. So how do you only fit one network by averaging over different fMRI at different time points and do the perturbation, or you also consider the time varying function itself?
No, that's a really good question. So obviously there's no fitting here, which I know is different than the rest of the talks, but this is something that people have done. So if you want to use one of these neural mass models, the intrinsic oscillations that you get are usually faster than the relevant time scale in fMRI. But what you can do first is you can apply the hemodynamic response function to try to simulate bold activity from the output of your neural mass model. And then once you have that, you're absolutely right that you can do more than just fit time averaged functional connectivity. You can also try to fit the statistics of time varying functional connectivity. And that's definitely something that people have done in these networked neural mass models. So you can, a simple approach is to just use a sliding window method on your data and obtain certain statistics about the dynamics of functional connectivity, and then try to match those in the model as well. And so I think that you're right that taking into consideration the time dependence of state is also definitely important.
But in that case, just one more question following up. In that case, are you going to still use the same neurodynamic function you currently have, or it's going to be some different dynamic functions that allows the time varying like change?
Right. So in what I presented when I was showing like the interareal coherence matrices, that is by definition something that's measured over a certain window of time. But even in the model, you can ask if the model itself exhibits fluctuating levels of coherence between different pairs of brain areas. So you don't need to add too much more complication in order to get the model to exhibit time varying coherence or functional connectivity. It's not, you can get that even without adding a lot of extra detail into the model.
Okay, I see. Okay, thank you for the answer.